Hello. My name is Dr. François Ricard. I am an osteopath and a PhD. I am director of the International Madrid School of Osteopathy, EOM, and president of the Scientific European Federation of Osteopaths, SCFO. In this video, I want to talk to you about the neurophysiological effects of spinal manipulation. This series of videos will look at the scientific literature surrounding the mechanisms of spinal manipulations, mostly used by osteopaths and chiropractors. In these videos, we will study the mechanical mechanisms of action in disc hernias, and the effects they have on the peripheral and central nervous systems, the neuromuscular, biological, neurovegetative systems, and on the organs. In this video, we are reviewing the neurophysiological effects of spinal manipulation. General points on the effects of HVT. 𝐸𝐭𝐫𝐢𝐜𝐻 et al. 2004 found that spinal manipulation stimulates spinal nerve root responses that were temporally related to the onset of vertebral motion. This photograph represents the experimental setup, according to 𝐸𝐭𝐫𝐢𝐜𝐻 et al. 2004, showing the application of a spinal manipulative HVT delivered with a mechanical force, manually assisted adjusting instrument adjacent to the bone pin, and accelerometer mount and spinal nerve root electrodes. According to George et al. 2006, local dorsal horn-mediated inhibition of C fiber input is a potential hypogesic mechanism of spinal manipulation. The graph on the left shows illustration of the temporal summation hypoalgesia for stationary bicycle, lumbar extension, and spinal manipulation. Picard and Bolton, 2012, proposed that spinal manipulation may produce a sustained change in the synaptic efficacy of central neurons by evoking a high-frequency, bursting discharge from several types of dynamically sensitive, mechanosensitive paraspinal primary afferent neurons. This illustration shows trunk mechanical stimuli responses of a single thalamic neuron in the ventrolateral nucleus according to Reed et al. 2000. Raw electrophysiological recordings of responses to lumbar trunk electronic von Frey stimuli, upper row, in the dorsal ventral thalamic neuron. A 45 degrees caudal, 73.9 grams. B 45 degrees cranial. C. Directions. D. The location of the trunk-responsive ventrolateral neuron was at the -2.5 mm caudal to bregma level. The shading indicates the search area. According to 𝐸𝐻𝐻 et al. 2016, short-term spinal manipulation does not affect the sensory motor functions. This picture illustrates the sudden load setup according to 𝐸𝐻𝐻 et al. 2016. The end stop describes the position of the weight when it is held by the participants. The drop height describes the position at which the weight is dropped. This model, according to Bielowski et al. 2009, provides visualization of potential individual mechanisms of manual therapy that the current literature offers as pertinent, and provides a framework for the consideration of the potential interaction between these individual mechanisms. Specifically, this model suggests that a mechanical force from manual therapy initiates a cascade of neurophysiological responses from the peripheral and central nervous system. Here we can see a table summarizing the classification of receptive nerve endings and their corresponding nerve fibers, according to Besson and Chouch, 1987. Spinal manipulation stimulates the neuromuscular spindle afferents, Group 1A afferents, and possible Group 2 afferents. Group 1 and 2 proprioceptive afferents. According to 𝐸𝐻𝐻, 1975, spinal manipulation increases joint mobility, applying a series of stimulations to the neuromuscular spindle afferents and small diameter fiber afferents, which silence the facilitated gamma motor neurons. The number of gamma motor neuron discharges is higher in the muscles associated with the segment of the spine that responds to the spinal manipulation. This is Picard's theoretical model, 2002, describing the relationships between spinal manipulation, segmental biomechanics, the nervous system, and end organ physiology. The findings of Picard and Wheeler's study, 2001, showed that spinal manipulation changes the discharge of Group 1 and 1 afferents. The neuromuscular spindles and the Golgi tendon organs of the multifidus and longissimus muscles are not stimulated in rest during a spinal manipulation, but are more stimulated by the thrust applied as part of the manipulation than by the static preload of the thrust. Their silence continued until the end of the manipulation. The picture on the right shows the sensory pathways that can change the discharge of gamma motor neurons, according to Picard and Wheeler, 1999. This picture, according to Picard and Wheeler's study, 2001, shows the original data from a subject with spontaneous paraspinal muscle activity in the lower lumbar spine, and its response to spinal manipulation activity, top trace, decreased in response to spinal manipulation. A tripolar, disposable, adhesive EMG electrode was placed approximately 2 to 3 centimeters to the right of the L4 spinous process, over paraspinal muscle that was taut when palpated. Bottom trace is the output from an accelerometer attached to the head of the activator adjusting instrument. Large accelerometer spikes represent the onset of each spinal manipulative impulse. Spinal manipulation was applied 3 times to the sacral base, third manipulation not obtained from the accelerometer, and successively applied to the L5, L4 and L3 mammillary processes. The large spikes in the top trace are likely mechanical artifacts from the manipulative impulse. Group 3 and 4 afferents. Picard and McLean, 1995, recorded facet capsule afferents during a spinal manipulation, responding in a graded fashion relative to the direction of force applied. The studies cited above make it reasonable to think that spinal manipulation may add a novel sensory input, or remove a source of aberrant input. This was confirmed by other Picard study, 2002. According to Gillette, 1987, spinal manipulation stimulates mechanoreceptors and proprioceptors, neuromuscular spindles, primary and secondary endings, and Golgi tendon organs. It stimulates low-threshold mechanisms, high-threshold mechanoreceptors, and high-threshold polymodal nociceptors. Therefore, all sensory neuron classifications, Group 1A, 1B, 2, 3, and 4 fibers could, in theory, be affected by spinal manipulation. Neuromuscular spindle stimulation. Picard and McLean, 1995, showed that graded sensory input from tissues surrounding the facet joint could elicit reflex muscle responses, similar to those measured by Indal et al., 1997. According to 𝐻𝐿 et al., 2013, spinal manipulations produce sustained changes to peripheral sensory signaling because of altered load transmission to the local mechanical environment. Thrust displacements produce sustained increases in the resting discharge of paraspinal muscle spindles. Reed et al. 2015. In the cat showed that neuromuscular spindle responses reduced significantly. This animal study showed that contact site for a spinal manipulation with HVT can have a significant effect on the magnitude of sensory input arising from muscle spindles in the back. The picture on the right is showing schematic representation of the preparation for delivering spinal manipulation to the lumbar spine, while recording sensory input from the dorsal roots. The motor was controlled by a programmable electronic feedback system, not shown. Tissues of the vertebrae receiving a spinal manipulation, L6 and L7, remained intact including the skin. Rotary motion of the motor's lever arm was converted to linear motion by a custom-built converter. The converter, in turn, was attached to a rod terminating in a small, circular, plastic tip of 5 mm diameter, placed on the intact skin at the contact site. According to 𝐩𝐚𝐫𝐽 et al. 1993 and 2000, changes in neuromuscular spindle input, produced by a spinal manipulation, can also contribute to the inhibition of somatosomatic reflexes. The picture shows the placement of the stimulator and the recording electrodes used to obtain cortical evoked potentials on magnetic stimulation of paraspinal muscles. According to 𝐸𝐻, 1975, spinal manipulation is responsible for producing impulses from gamma neuromuscular spindle afferents, as well as small diameter fiber afferents, as well as silencing the gamma motoneurons, and the neurons that innervate the paravertebral tissue, group 1 and 2 proprioceptive afferents. This picture modified from 𝐻𝐻𝐿 and 𝐻𝐻𝐿, 1995, shows that inhibition of alpha and gamma motoneurons is caused by stretching of the neuromuscular spindle and stimulation of the Golgi tendon receptors. From the Picard and Wheeler's study, 2001, this picture shows that original response of a neuromuscular spindle to a similar load to a spinal manipulation. Original tracing of muscle spindle's response to spinal manipulative-like load. Inset shows spindle's discharge on expanded time scale immediately before, during, and shortly after impulse. The effects of spinal manipulation on the higher neurological centers. According to 𝐻𝐻𝐽 et al. 2000, spinal manipulation affects cerebral evoked potentials from paraspinal muscles, which suggests that the increased sensory input of paraspinal neuromuscular spindles during a muscle spasm could contribute to the reduced amplitude of cerebral evoked potentials. The study by Reed et al. 2014 examined the effect of the duration of the thrust during an HVT manipulation on lateral thalamic thresholds in response to specific nociceptors. The duration of the thrust during a spinal manipulation does not affect brainstem thresholds. This figure shows electrophysiological recordings, the lower line, of responses to electronic stimuli applied to the lumbar spine, the upper line, in the ventral posterior nucleus. This diagram shows the trunk mechanical stimuli responses of a single thalamic neuron in the ventrolateral nucleus, according to Reed et al. 2000. Raw electrophysiological recordings, lower row, of responses to lumbar trunk electronic von Frey stimuli, upper row, in the dorsal ventral thalamic neuron. A 45 degrees caudal, 73.9 grams. B 45 degrees cranial. C directions. D the location of the trunk-responsive ventrolateral neuron was at the -2.5 mm caudal to bregma level. The shading indicates the search area. According to 𝐸 et al. 2014, stimulation of the lumbar spinous processes revealed bilateral neural responses in medial parts of the postcentral gyrus, S1, the secondary somatosensory cortex, S2, the posterior parts of the insular cortex, and the cerebellum. Thumb stimulations revealed activation only in lateral parts of the contralateral S1. This study demonstrates that the application of posterior-anterior pressure on lumbar spinous processes produces neuroplastic changes in subjects with chronic low back pain. The photo on the left shows that stimulation triggers well-defined brain activity in the primary somatosensory cortex, S1, because of lumbar stimulation. The photo on the right shows the results from a whole brain analysis during stimulation of the lumbar spinous process, according to 𝐸 et al. Reed et al. 2014 showed that neuromuscular spindle responses increased significantly during a spinal manipulation with HVT. The numbers represent the number of afferents with their receptive field in this location. A shows the dorsal view, and B shows a cross-sectional view roughly aligned with dorsal view. According to Peltier et al. 2018, the neurophysiological effects of osteopathic manipulative treatment may help to normalize the properties and organization in cortical sensory motor areas, and impact the autonomic nervous system. The results of the study by Ponzo et al. 2018 provide support for the effects of osteopathic manipulation on cortical plasticity. Conclusions. Spinal manipulations with HVT seem to act on paraspinal proprioceptive receptors, group 1 and 2, and 3 and 4 afferents. Neuromuscular spindles of the paravertebral muscles. Upper neurological centers, cerebral cortex, thalamus, autonomic nervous system, etc., and they modify cortical plasticity. In the next presentations, we will look at the effect of osteopathic treatment on central and peripheral sensitization, and the autonomic nervous system. Here you can see a list of all the references used for this presentation. Thank you for listening.